Hi, I'm Trent Northen from Berkeley Lab, and it's my pleasure to give you a little bit of background on this conference and also to tell you a little bit about what I'll be covering in my talk. So I'm very excited about the Copenhagen Bioscience Conference on Plant-microbe Interactions for a few reasons. The first is that it brings together leading labs from around the world in studying plant-microbe interactions. Many of the groups that I follow closely. The second is that this is going to be a very relatively small conference, just maybe 150 attendees. So this will give us all chances to get to know each other and talk about science, which is something that I've been missing very much over the last few years, and I'm guessing you have been too. And then finally, I think it's just fantastic that the Novo Nordisk Foundation is sponsoring this conference, and so that the attendees only have to pay for their travel expenses. And I think this will help make for a really inclusive conference. So I think that's another really nice thing about this. In terms of my talk, our lab is really interested in studying the role of metabolites in mediating plant microbe interactions. And so I'll start off by telling you about a standardized grass rhizosphere community that we've built and some fabricated ecosystem devices for studying plant microbe interactions. And then I'll tell you about an ongoing study that we've been working on, studying the role of stresses in mediating root exudates and how this changes the composition of the exudates. And in turn, how these metabolites that are upregulated in response to stress can select for microbes that can help mitigate plant stress. And so this provides a mechanism by which the plant can help control its microbiome, especially when it really needs the microbes to help address challenges in the environment. And also that we found some of these metabolites that are being upregulated in response to stress can promote root growth. And then I'm going to tell you about how we've been using metabolomics to study plant use of organics directly from the environment. So it's not just microbes that can use the exogenous metabolites, it's also plants. And so we used a diverse panel of plants to study the use of organics across the diel cycle. And we find that the profile of organics changes depending on whether it's night or day, and that depending on the type of plant, they use different profiles of metabolites from their environment, that this can be a significant part of the total carbon budget for the plant, and that some of the metabolites that the plants use actually promote plant growth. And this isn't just a nutrient effect. They actually have some unknown mechanisms for promoting plant root growth. Then I'm going to switch over and tell you about some of our technical developments in building what we're calling the EcoBot. So this is an automated system for studying plant microbe interactions in the rhizosphere. And we were integrating this with AI to enable a self-driving system. That's the eventual goal, at least, to create a self-driving lab for the environment that can not only help us understand rhizosphere dynamics and assembly processes and stress responses, but can help us optimize microbiomes for sustainable agriculture. So with that, those are just some of the topics I'm going to cover, and I'm very much looking forward to talking to you and meeting you at this conference. See you there. Thanks.